And I would also like to introduce now our last speaker for the first session, who is Romain Veltz. He's a researcher in mathematical and computational neuroscience at the INRIA Centre at Université Côte d'Azur as a part of the Chronos team. He's currently focusing on describing and analyzing neural networks from the perspective of dynamical systems and probability theory. His research areas include tractography, whole brain models, neural plasticity, spiking neural networks and their main field, and signal integration in dendritic spines and synapses. So this is more an in silico presentation now. I'm really curious to hear what you are talking about. The virtual stage is yours, Romain.
Thank you very much for the kind introduction. And effectively, we will be going now into in silico, let's say, models. And so I would like to present you SynPlas tool. SynPlas tool is a project for which is basically the development of a graphical user interface for the prediction of synaptic plasticity outcomes in silico. And so the coordinator of the project is Hélène Marie. She's a neuroscientist in the south of France at CNRS, specialized in synaptic plasticity, Alzheimer's disease. And I collaborate with her. And I am from INRIA research team. So the main point about this is basically studying synaptic plasticity. And why is it important? To me, I think it's really important. But synaptic plasticity, for those who don't know, it refers to the ability of synapses, so the connection between the neurons, to change their strength and efficacy in response to activity and experience. So it is a fundamental mechanism underlying learning, memory, and adaptation in the brain. So these connections, the synapse, they strengthen or weaken based on the frequency, intensity of the signals, dictating the encoding and retention of information. And so how you stimulate the presynaptic neuron and the postsynaptic neuron, and how you do that in relation with one another, dictates how the synapse behave. And so discovering the rules of synaptic plasticity, meaning what are the conditions, for example, the stimuli that imply a change in synaptic plasticity. So this is rules of synaptic plasticity. So discovering these rules is fundamental for a better understanding of how the brain works, and, for example, to identify pathological mechanism behind various neurological conditions. So, for example, deficits in synaptic plasticity have been implicated in conditions like Alzheimer's disease, Parkinson's disease, schizophrenia, and others. And Hélène Marie's lab focuses a lot on Alzheimer's disease and the study of synaptic plasticity in Alzheimer's disease. And at first, the model of synaptic plasticity that I will be presenting, we wanted to model Alzheimer's disease with Hélène, and basically we failed in the time frame that we were given. We could only study synapses in, let's say, a working condition. And of course, one last application of synaptic plasticity is for machine learning and data science, where we all know that an excerpt of synaptic plasticity, let's say the Hebs rule, is used in neural networks, and the type of synaptic plasticity that I will describe is also used routinely in spiking neural networks. So what is a typical experiment? I recall that the goal is to know how to study how synapses adapt and alter in response to stimuli and intervention. So usually rat and mice are commonly used in experimental research to study synaptic plasticity, basically because they are very close to because of their genetic similarity to human, the ease of handling and relatively well understood neurobiology. And so what experimentalists usually do is to take rodent brain slices and perform electrophysiology of the synapse function. Now, for statistical significance, they must repeat the same experiment many times under a specific condition and in different mice. And so a typical day for Ellen Mai's lab is one animal that is sacrificed, and it gives 3 data points. And so we will see what it gives for synaptic plasticity protocol. So on the right side, you have here a typical brain slice, and here you can see the hippocampus. And so usually what we will be focusing is on a specific pathway in the hippocampus called the CA3-CA1 neuron. And so you have a stimulating electrode in the CA3 neuron, basically pathway that will excite the CA1 neuron, where we have a recording electrode. And so this is a protocol that is used by Dr. Mary's lab, but it's used in many labs as well for studying excitatory synaptic plasticity. And so basically, Ellen told me that they are using a few hundred mice per year in our lab for these experiments. A typical experiments now amounts. So I want to give you a glimpse about the complexity of this mechanism and why it requires a lot of experiments and so animal studies. So a typical, so if I take the same color code here, so I stimulate the CA3 neurons. So you can see here I stimulate them at around 10 hertz, 300 times, and I record in the, let's say the blue neuron, I record the response of the stimulation. So after the stimulus has been done, so the high frequency stimulation, I can record. I'm going to stop that. I can record the synapse efficacy. And what is amazing, but that is done from the 70s, is that the synapse efficacy can strengthen. So in this case, it's about 1.5 times the efficacy it had before. And this efficacy outlasts protein turnover. So in this case, it lasts 50 minutes. So this specific protocol induces a potentiation of the synapse. Okay. But now we have, that's a type of plasticity rule, okay? We have a diversity of rules. And so, for example, here in this little schematic, if I stimulate the red neurons once and the blue neurons twice, and I repeat this whole thing 100 times, then I can induce, again, an increase in the synapse efficacy, so potentiation of the synapse. And what is very amazing and difficult from a modeling point of view, or just to study this system, is that if I only stimulate once the presynaptic neuron and once the postsynaptic neurons, okay, and I repeat this 300 times, I have no change in synaptic efficacy, okay? And so if we do that for many protocols, so basically, oops, sorry, I can change the time in between, let's say, the presynaptic stimulus and the postsynaptic stimulus. I can change the frequency and so on and so forth. I have several protocols that were studied in Tigare in 2016. And so here you have the connection change. So basically when it's above this line, we have an increase in synapse efficacy, and it's below we have a decrease in synapse efficacy. So for, let's say, this protocol, 2 post stimuli and 1 presynaptic stimulus, separated by 50 milliseconds, sorry. Oops. We have what I want to show you, what I want to underline, sorry, we have a distribution of responses. Okay, so each point here is a data point, so it's basically one animal. And you can see that sometimes we have a depression of the, synaptic depression, or sometimes you have synaptic potentiation. For this protocol, it's mostly synaptic depression. For this protocol, I have a problem. And for this protocol, it's mostly synaptic potentiation. But so we have various protocols, and for each protocols, the synapse has a noisy behavior. So we need to repeat the same protocol many times in order to extract the distribution. So you see that combined with the minute changes in the stimuli and the noisy behavior, this makes it a very difficult system to analyze and to study. And just to name a few, so that was the paper by Tiger et al. But we can change the presynaptic frequency, we can change the ratio of, let's say, concentration, the concentration of magnesium and calcium, and this affects the plasticity rules. It depends on the temperature, on the age, so on the age, on the ions concentration, and so many animals are sacrificed, and it's a total mess from the point of view of theory, from a theoretical point of view, because if we want to abstract a rule, it's very difficult to find, let's say, a principle underlying those change and behaviors. So let's dive now into the model. And so the computational modeling, it allows basically to for experiments to be carried out much more quickly. We will come back to this, but more or less the simulation is time is 1-to-1 with stimulation time. So 1 second in real time is 1 second of computation. And so this model reproduces experiment, but it makes a lot of predictions. So for example, it allows to save time and also to and animals. It also computational modeling allows to select a subpart of interesting, so to say, experiments if the model, of course, is well tuned and well characterized. And for me, what is more important, it allows abstraction, so finding principles behind the experiments. So some to say, can we have a toy model behind those facts? And also, once we have a working model for non-pathological synapse, we can study diseases and pathologies such as Alzheimer's disease. And so what I will present is basically a model that we published with Hélène and co-workers about 2 years ago. And we were able to reproduce 9 different rules, or let's say experimental papers, with a single model by changing what we call external parameter, temperature, age, ion concentrations, stimuli. And so with a single model, we are able to reproduce all these facts. So how does it work? One big issue, and I told you, I mentioned this before, the synapse behavior is noisy. And so here you have, on this little schematic on the left, you have a glutamatergic synapse that is depicted. So you have the presynaptic, let's say, compartment, and then you have the postsynaptic side of the spine. On the presynaptic side, we have vesicles, and so population of vesicles containing neurotransmitters. These are models with Markov chains. And on the postsynaptic side, we have AMPA receptor, which are very fast. We have NMDA receptor, voltage-gated calcium channels, sorry, small potassium channels. And so these are these are models with Markov chains, okay? And so it was quite difficult because those Markov chains have to be tuned to different temperature to reproduce experiments. But so if I run, let's say, a presynaptic stimuli, a spike is coming onto this synapse, then it will trigger release of neurotransmitter and opening and close activation of these different receptors. You have a graph of these opening and closing of the different receptors. So for example, the VGCC, you can see that this type open and close all the time. If you look at NMDA receptor, it's the same, it opens, it closes, it opens and closes. So it's obvious from this slide that if you give the same presynaptic stimulus, you have a diversity of responses, basically a random or noisy responses. And so that's one big issue. It means we have to repeat numerically a lot of experiments in order to extract distributions. But needless to say, to give you a glimpse about how the type of result that we have, on the left, we have the data points from Tigare et al. I was just describing before. And on the right, you have the distribution of responses that is provided by the model. And so you can see that we manage, except in these cases, maybe, we manage to capture the distribution of responses given by the experimental data. Okay, and so that's one experiment out of the let's say the many that we describe in the paper. But more or less, more importantly, we can make prediction as well with this. And so, for example, here we can change the number. So we had the same stimulus repeated 300 times, but if we change the number of repetitions, so let's say 100, 300, or 700 times, we can make prediction. And so in this case, for example, we can induce, let's say, more synaptic depression if you increase the repetition, or in this case, for this pre-post delay, we will induce more synaptic potentiation. And so, idem, if we change temperature, magnesium, calcium, or age and frequencies, this is all taken into account by the model. The proposal, so to remind you, is to predict synapse plasticity with a large panel of options to vary experimental conditions. So the open source code is available freely on GitHub. It's written in a quite fast language, and so basically 1 minute protocol is simulated roughly with 1 minute. And so what we aim to do is to provide a user-friendly web interface to obtain a measure of synapse plasticity when providing, so by the user, the experimentalist provides experimental variables that they want to try. And so we want to provide a graphical user interface, so the experimentalist doesn't have to, or the user doesn't have to program in C++ or in Julia. We will provide documentation and tutorials and videos. We have collaboration with the private company MediaVince for the web development. And at this time, we are not certain whether it will be a web application, so people just logging into a website and run the computation on the website and get the answer, or a standalone application. Okay. And also, we need to enable parallel computations for repetition of the same stimulus, because we saw that the synapse response is highly stochastic. So we have two proof of concept right now. Here you can see a very crude proof of concept for web interface. So we can have the stimulation protocol, the stimulation paper, the protocol here, 1 pre, 2 post 15 in this case. We can put the age of the mouse, the temperature, the calcium, and then we analyze the parameter. And so we make a simulation and we see the synapse efficacy that is displayed to the user. We have another one. I want to show you this. In video. So you have a menu. So this one is a standalone application. So you can select the different paper and we can run the simulation. And you can see that for the particular protocol, here you have, for example, the activation of 2 major proteins involved in synaptic efficacy, and the weight change is displayed on the last panel here. And so the goal is by changing these box and buttons, the user will be able to run these experiments and select what experiment they want to perform on animals. And so the conclusion, 2 master students will be hired to, so we have proofs of concept, and now we have to implement this more thoroughly. And so we have 2 master students that will be hired for for next year, basically in February. And we had some difficulty to hire them because we wanted a specific profile, basically at the interface between modeling, neuroscience, but also computer science, and need to be good at programming and web development and GUI development, but they need to understand what they're doing math-wise and biologically-wise. And that's why it was quite difficult to select these people, but now these students are found. And so with this, I'll thank you for your attention. And if you have any questions, I'm happy to answer, try to answer. Thank you.
Thank you very much. Thank you very much, Romain, for this more in silico view of these models. Are there any questions from the chat?
There were indeed a few questions. One question, for example, was relating to the previous talks with the diseases, if you are able to model effects of mutations on the channels.
So we are able to do that. And so in effect, you will need to tell us the mutation. And so basically change the Markov chain describing this channel. And then it will be the model will be able to produce the activation of the different proteins. But I mean, this is quite difficult to do because usually you need to do that with data to be sure that the model is doing at least reproduce some non-experimental data. But yes, that was actually our goal. We wanted to study synaptic plasticity in Alzheimer's disease, but it took us so long already to get it working, so we didn't do it.
There were some special questions, like if you can reproduce a BCM rule, or if you can also produce plasticity such as TBS, LFS, and HFS with your models.
I don't think we tried the BCM. I don't think we tried the BCM rule inside. It's a good question. We tried something that is not too far, Dudek and Bear experiments, in which we varied the frequency of the stimulus, and we could reproduce the basically no change, depression, and potentiation. And so the difficulty was to be able to reproduce rules that do not depend on precise spike timing, and those who really depends on precise spike timing, that the one of Tigare that I described. Is your model based on physical detailed cells? Just to know whether it's possible. Okay. Oh yes, that's a question that you asked, but there are 2 parts. I don't know if can you see my slide now, or can you see?
Yes. Yeah.
So I forgot to mention that there are 2 layers in the model. So there is one layer which is very biologically relevant, meaning that we describe with a lot of details the channels. What is less known, at least biologically, is the behavior or the equations for modeling calcineurin and CAMK2. And so we have a model, that the latest model that is used for those proteins, but we do not use Markov chain to model those. And we use, let's say, deterministic equation for that. So there are 2 layers of complexity, or let's say biologically relevance in the model. The first layer is quite well modeled, and the second layer, which is less known, is not modeled in the same way. So I hope it answers for the one question in the chat. And so,
okay.
And the classical stimulation, stimulation is very easy to simulate. You just provide the pre and post spikes, and so theta burst stimulation and classical high frequency stimulation are very easy to do. Like high frequency stimulation is like the Bliss and Lomo protocol. So yeah, that's possible.
Okay, then I think for the sake of time, we have to close here. Thank you very much again, Romain.